Hello everyone, welcome back for more biotechnology. This is going to be the last video in this chapter, recent advances in medical biotechnology. Now there's a lot of different things that the book chooses to talk about here. For this video, I'm going to focus in on one particular one, which is the use of biomarkers. So let's go ahead and start talking about that. So ultimately, the goal of diagnostic medicine is to definitively tell the difference between someone who has a disease and someone who does not. Now, this is pretty obvious to us because we have a very good and very unique example in the past year, the COVID test, right? So when the COVID virus, the SARS-CoV-2 coronavirus, made its way over here to America back in March, we early on didn't have a really good way of telling who has the virus and who doesn't, right? Not until we developed a diagnostic test. So these are the sorts of things that we really want to focus on here, and it is obviously of critical importance to be able to tell who is sick and who is not, because that is ultimately going to guide who are we going to treat and how are we going to treat them without doing them undue harm. So biomarkers are characteristics that help us to make these sorts of determinations. Now, biomarkers come in a number of different forms and fashions. Now, biomarkers can be particular proteins, like what you're looking at here in this picture. We're actually looking at a new technique that we haven't talked about here that also makes use of antibodies. This is called immunohistochemistry, or IHC. What we're seeing here is a section of tissue, particularly from breast tissue, and we have exposed this tissue to an antibody against a particular antigen that is only produced by cancer cells. So what we're seeing here in this dark brown color is a positive reaction to the antibody. So we can use this sort of test as a definitive test for whether someone has breast cancer or not. So this protein would be an example of what we call a biomarker. So biomarkers certainly can be proteins like what we see here, but a biomarker can be any sort of molecule that we can measure, things that are in bodily fluids or blood or urine, things that are easily measured and easily quantified and clearly easily detected. Or, as we're going to see on the next slide, biomarkers can be actual DNA sequences. We've already seen, and we're going to continue to see, that certain diseases are characterized by genetic mutations. So by doing genetic screens, we can determine who has those mutations and who does not. And ultimately, in that case, the DNA sequence actually itself becomes a biomarker. So if you're talking about genetic screening as biomarkers, then we need to become familiar with what's called a pedigree chart, which is something that you often see in an introductory college biology class. So the pedigree chart, like what you're seeing here for the disease cystic fibrosis, will track the inheritance of disease-causing genes from generation to generation. And other information in the pedigree chart allows us to know who has the disease, who does not have the disease, and who is a carrier for the disease. Now, a reminder, because it's easy to forget, every cell in your body except for your gametes, your sex cells, either sperm or egg, have 2 copies of each chromosome. So you've got 2 copies of chromosome 1, 2 copies of chromosome 2, and so on and so forth. So, therefore, you have 2 copies of every gene. So, if we know that a disease is caused by a mutated gene, we need to know whether that mutation exists on one of the chromosomes or both of the chromosomes. Now, genes can naturally vary from chromosome to chromosome, and these gene variants are called alleles. Having different combinations of alleles gives us our different physical characteristic traits, like our hair color, our eye color, how tall we are, the shade of skin that we've got working for us. So having different gene variants, these alleles, is actually fairly normal. But some alleles are severely mutated and they do cause disease, like what we're seeing here in cystic fibrosis. So in the case of cystic fibrosis, a mutation to a gene called CFTR causes the protein product of that gene to not be functional. It just barely gets made and then it gets destroyed by the cell, so it's not able to carry out its protein duty. But if that one mutated bad allele is the only bad allele that you've got, meaning that your other allele on the other chromosome is actually good, you actually do not have the disease. You are what we call a carrier for the disease. In order to actually have full-blown cystic fibrosis, you've got to have bad alleles on both chromosomes. And obviously, if you have two good alleles, you're all clear, right? So if you look at the pedigree chart here, if you look at the top generation, which would be the grandparent generation, and then you have their kids down here and then their grandkids down here, the grandmother, the affected female, must have two bad alleles, which gives her cystic fibrosis. And then her husband, the grandfather, has two good alleles because his square is shaded in white. Now, because she has two bad alleles and he has two good alleles, all of their children are guaranteed to be carriers because she's going to give one of her bad alleles and he's going to give one of his good alleles. So this is why they are shown in this kind of light green color. They are symptomless because they are carriers. They can get away with having one good allele and just one bad allele. Now, if you'll notice, one of their children, this mother right here, her husband is also a carrier for the disease. So if you do the math, they have a 25% chance of one of their children having full-blown cystic fibrosis, a 25% chance of having a child who is totally normal, and then a 50% chance of having children that are carriers themselves. And since they have 4 children, that is bared out here, although that's not necessarily always going to be the case. Just as the case is, just because you flip a coin 2 times doesn't necessarily mean you're going to get one head and one tail. So the important information to take away from this is that being able to identify genetic biomarkers allows us to track the transmission of genes from generation to generation so that we know the statistics behind, okay, if we have these 2 people here, this man and this woman, if they have children, this is the chance that they're going to have this disease. So the earlier you can start counseling them on that in terms of what they can expect, the better, obviously. So this pedigree chart that you're seeing right here is much, much, much more complicated. So this is a multi-genetic, multifactorial pedigree chart, but we're not going to get too heavy into it, but the ultimate takeaway here is that genetic biomarkers are very often used for cancer screening. So for example, one of the most commonly mutated genes in breast cancer and colorectal cancer in women is a gene called BRCA1. So for a pedigree chart like this, we would obviously want to, for say a woman that has a history of breast cancer in her family, we might want to subject her to a genetic screening for the BRCA1 gene to determine what exactly her risk is for developing breast or colorectal cancer herself. So once again, she can be counseled in the right way, again, so she can know what she can expect, and she at that point would know maybe she needs to have mammograms done on a more frequent basis. So basically the big takeaway here is that because our understanding of genetic inheritance is already very, very, very good, there's not a whole lot we don't know about that at this point. The idea is that if we can develop more genetic biomarkers, then our ability to treat diseases earlier is only going to get better and better. All right, so that is going to do it for Chapter 12. I certainly had a good time talking to you. I will see you next time for Chapter 13. See you later.